Bioinfocongress II Dr. Senkal Bioactive Sphingolipid Metabolismasının düzenlenmesi. Dr. Senkal, lisans eğitimini Güney Carolina Tıp Üniversitesi'nde tamamlamıştır. Doktor sonrası çalışmalarını New York, Stony Brook Üniversitesi'nde tamamladıktan sonra Virginia Commonwealth Üniversitesi'nde Biyokimya ve Moleküler Biyoloji bölümünde bağımsız araştırma programına başlamıştır. Ve burada sphingolipidlerin kanserdeki metabolizması, düzenlenmesi ve biyolojik işlevlerini araştırmıştır. Dr. Senkal'ın laboratuvarı NIH'den Merit R37 araştırma hibesi ve VCU'daki Massey Kanser Merkezi aracılığıyla kanser araştırmalarında Harrison bursu tarafından finanse edilmektedir.
Bioinfocongress II Dr. Senkal Regulation of Bioactive Sphingolipid Metabolism. Dr. Senkal finished his undergraduate education at the Department of Biological Science in Middle East Technical University, METU. He then moved to Charleston, SC, for his graduate training at the Medical University of South Carolina. After completing his postdoctoral studies in Stony Brook University, NY, Dr. Senkal started his independent research program in the Department of Biochemistry and Molecular Biology at Virginia Commonwealth University, VCU, where he studies the metabolism, regulation, and biological functions of sphingolipids in cancer. Dr. Senkal's laboratory is founded by Merit R37 research grant from NCI, NIH, and Harrison Scholarship in Cancer Research through the Massey Cancer Center at VCU.
Sayın Dr. Senkal ve değerli katılımcılarımız, Bioinfocongress II etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün Regulation of Bioactive Sphingolipid Metabolism konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüz olacaktır. Merhaba Can Hocam, hoş geldiniz.
Merhabalar, hoş bulduk.
Dilerseniz sununuza başlayabilirsiniz.
Peki, teşekkür ederim. Ben artık başlıyorum sizle izleyenlere ve tüm Bioinfocongress çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz, zamanınızı ayırdınız. Şimdi sizlere bizim laboratuvarımızda yapılan bazı şeylerden bahsedecektim. Yeni yapılanlardan, ancak telif hakları nedeniyle bunu yayınlamamız mümkün olmadığı için konuşmamı birazcık değiştirmek zorunda kaldım. Ama umarım sizler beğenirsiniz. İşte ise bir şeyler öğrenmiş olursunuz bu sphingolipidlerle ilgili. Bir de birçok terimin Türkçesini bilmediğim için sizlere hep İngilizce anlatacağım. Sorularınızı Türkçe, İngilizce karışık nasıl isterseniz sorabilirsiniz. Bununla birlikte başlayalım. Şimdi size işte Sphingolipid Metabolism and the discovery of a novel ceramide metabolic pathway. I'll tell you about this. So here is this is my outline for the talk, and our objectives would be learning about the sphingolipids and the pathway that we were able to identify. So I'll tell you a little bit about the historical perspective of how sphingolipids were discovered. And then we need to talk about the sphingolipid structures before we can go and talk about the metabolism. And then we will briefly touch on the intracellular compartmentalization of the metabolism and also signaling. And then I'll tell you just a bit about the functions of sphingolipids. These are going to be in general, just one slide. And then I'll tell you about some of the previous findings from my group, from me, and when I was in Stony Brook University particularly. And we will conclude, and I will get your questions, and I'll try to answer your questions. So historically, initially the sphingolipids were identified by a gentleman called Tuticum in the brain isolate. So this was done in the 1980s. So back in the day, chemistry was still the hot topic, and people were able to extract many things from tissues. And this is what he did. He extracted a certain kind of lipid that he knew it was a lipid, but he didn't know what it was. And at the time, this was a very big riddle. It was a really enigmatic lipid. And he was influenced by the Greek mythology. And as many of you or some of you would know, that in the Greek mythology, there is Sphinx. So Sphinx asks this question, what creature is on 4 feet in the morning, 2 feet at noon, and 3 feet in the evening? So obviously this is an easy question for us to answer now, but the enigma was still there. So he named this class of lipid that he isolated as sphingolipid. So I hope you know the answer to this question meanwhile. So here is the structure. So first of all, we have to clarify that for all sphingolipids, for a lipid to be classified as a sphingolipid, it has to have the sphingosine backbone structure. And here is the sphingosine backbone structure. It has 18 carbons. And it has this amino alcohol group. And first is the alcohol group, and the second carbon is an amino group. So classically, this is D-erythro-2S3R4E sphingosine. So this is the chiral center on the second carbon. So it is S configuration, and the third carbon, it is R configuration. And there is a double bond in 4E. This is cis, so that's E, and it is 4E sphingosine. As I said, for any sphingolipid, any lipid to be classified as a sphingolipid, this backbone has to be present on. So here is maybe this one is on the left is a comparison of a sphingolipid very abundant. This is sphingomyelin. So sphingomyelin has this sphingoid backbone, and there is a fatty acid chain attached to it, to its amino terminal, and there is a phosphocholine group. If you look at side by side with the phosphatidylcholine here, which is a phospholipid, a known phospholipid, there are certain similarities, but many differences at the same time. And as you can imagine, with all these chiral centers and double bonds, there can be many sphingolipids. So these are some of the examples that the sphingolipids can be in the nature. When we look at the abundance of these lipids, and this is from a paper from Ilya Leventhal in UVA. So I need to specify that this is plasma membrane, by the way. So they are looking at the plasma membrane composition of these lipids. So this SM sphingomyelin is mainly on the outer leaflet of the plasma membrane. And it is very comparable amount to the phosphatidylcholine. However, if you look at the ceramide, which is the main or the central molecule of the sphingolipid metabolism, the amounts are extremely low, which is why we think that these low amounts, once they are modulated just a little bit, they can have signaling functions, so that you have a really tight control on the amount of ceramide. So now let's talk about the biosynthesis of sphingolipids. So here I'm showing you the de novo synthesis, how we synthesize these lipids in the body. So it starts with the condensation of serine and palmitoyl-CoA by the action of this enzyme called serine palmitoyl transferase, which generates the beginnings of this fatty acid, the backbone, the sphingoid backbone. This is a triketosphinganin, which is reduced by the sphinganin reductase to generate the sphinganin backbone. So sphinganin is lacking this double bond. However, it can be utilized by our favorite enzyme, ceramide synthesis, to generate dihydroceramide. So the ceramide synthesis, they take this fatty acyl CoA and then they attach it to this N-terminus. So they N-acylate the sphinganin backbone. Then we end up with this compound. This is dihydroceramide. And once dihydroceramide is desaturated, then this double bond is introduced, and we have ceramide, which again I said this is the central molecule. And ceramide can be cleaved by the ceramidases, so at this amino group here, to liberate fatty acid and sphingosine again. And this sphingosine, once it is phosphorylated by sphingosine kinases, you see this one hydroxyl group is where the phosphate is added, then we generate sphingosine 1-phosphate. So sphingosine 1-phosphate has also signaling functions through its receptors as well on the plasma membrane. And then the only way for the sphingosine backbone to leave the metabolism is through the hydrolysis by the sphingosine 1-phosphate lyase, which generates the hexadecanal and ethanolamine phosphate. So here, once we have ceramide generated, this ceramide can also be converted to complex sphingolipids. And these complex sphingolipids can be sphingomyelin. I showed you the structure before, which is the addition of this phosphocholine on this one carbon, or glycosphingolipids, where the sugar moieties are added to this one first carbon. So now I'll tell you just a little bit more about the serine palmitoyl transferase. So this is the recently published structure of serine palmitoyl transferase. This is a multimeric protein, and it is located on the endoplasmic reticulum surface. So as I mentioned to you in my earlier slide that it uses serine, but in fact it uses primarily serine as the amino acid component for the sphingosine backbone. But it can also utilize alanine and glycine. And these can be very important depending on the context that we are talking about. So for our purposes, for my talk, we'll talk about just the regular sphingosine, which is the palmitoyl-CoA and serine combined together. So now let's talk a little bit more about the ceramide synthesis. And I am biased because for me, ceramide synthesis are the most important enzymes because of my research interest. So why are they important? So maybe I'll go back one slide and I'll tell you here, and then I'll mention more. So here, as I mentioned to you, ceramide synthesis attach this fatty acid chain into this N-terminus here, right? Or amide group here. So here, this is C16 ceramide because the fatty acid chain has 16 carbons to it. Very importantly, there are 6 ceramide syntheses from SIRS1 to SIRS6, and these are encoded by individual genes, so these are not just splice variants. And what they do is they have a preference towards the utilization of different fatty acid CoAs with different fatty acid chain lengths. What do I mean? So, for example, SIRS1 very specifically uses C18 fatty acid CoA to generate C18 ceramide with the dihydrosphingosine or sphingosine in the salvage pathway. On the other hand, for example, SIRS2 uses very long chain fatty acyl CoAs to make very long chain ceramides. And very importantly, these ceramides with different fatty acid chain lengths, so we call them different ceramide species, these species can have different signaling functions. So this becomes like a coding factory that the SIRS really determine what kind of ceramide needs to be made for what kind of signaling effect. So then let's talk about a little bit of the complex sphingolipids. So here I'm showing you the sphinganin backbone. With SIRS addition, we have the dihydroceramide. And then it is converted to ceramide where this double bond is added by the desaturase enzyme. Or sphingosine can also be used by the SIRS activate SIRS, and we have ceramide. So for sphingomyelin generation, we have the sphingomyelinase or sphingomyelin synthase. It goes this way, so it adds this phosphocholine group. On the other hand, sphingomyelinases can cleave this phosphatidylethanolamin to generate ceramide. On the other hand, as I mentioned to you, the glycosphingolipids, these are the sugar moieties that are added on the this 1-hydroxyl group of ceramide. So here are some of the examples. We have galactosyl ceramide, glucosyl ceramide. And galactosyl ceramide can give rise to the sulfatides, which are very important, especially in the neuronal functions. And also I should mention to you here that sphingomyelin, as the name would imply, this is one of the most abundant lipids in the myelin structure that is going around the axons of the brain mainly. And we think that Tuticum, when he extracted the lipids from the brain, he probably most likely extracted sphingomyelin because of its abundance. That's what we think. So I have been telling you about ceramide synthase using sphingosine or dihydrosphingosine. So the main thing is that we call this when the ceramide synthase uses sphingosine, it is really the recycling pathway where the complex sphingolipids can be degraded into sphingosine and the sphingosine can be reacylated. So this is what I mean. This is another slide for it. And so here is like another representation of the sphingolipid pathway. So it itself is very, very complex and very regulated, but at the same time, it is also highly compartmentalized in the cells. So here is how it is compartmentalized. The de novo pathway that I talked to you in detail occurs on the cytosolic phase of the endoplasmic reticulum, where all these enzymes are located here. So once we generate ceramide on the ER surface, this has to be transported to Golgi for its conversion to the complex sphingolipids, such as the glucosyl ceramide by the action of glucosyl ceramide synthase, or to SM by the action of sphingomyelin synthase. So very importantly, we now know that there is a protein called CERT, ceramide transport protein, that is also responsible for the transport of ceramide from ER to Golgi to be utilized for the sphingomyelin synthase for SM generation. So once we have these complex sphingolipids generated in the Golgi, then they end up in the secretory pathway, and they also end up on the plasma membrane. And on the plasma membrane, we know for sure that either through endocytosis and vesicular endocytosis, or through the actions of these other metabolic enzymes of the sphingolipid pathway, almost the whole cycle can be again reversed. And when the endocytosis happens, these complex sphingolipids end up in the lysosome, and then they are cleaved to generate sphingosine. And I mentioned to you that this sphingosine can feed back into the de novo pathway in a way to be used by ceramide synthesis. So this is the part that we call salvage pathway. So let's talk a little bit about the sphingolipid functions. as I told you, the sphingolipids are very important. So mostly or mainly they were initially identified because of their structural functions. So they are important in myelin, as I said, because of the SM presence. And also sphingomyelin is also known to be coupled to cholesterol on the plasma membrane as well. So it has some functions in regulating the abundance of other lipids. And at the same time, sphingolipids have biophysical properties. These are unique properties because of the amino group there that can make other hydrogen bonding on the membrane surface that can create the membrane curvature, and they can change the lipid packing of the membranes. There is some ideas about the membrane raft formation that can be involved in the receptor capping on the plasma membrane. We also know that they can act as the toxin binding sites. For example, cholera toxin needs to bind to the GM1, which is a sphingolipid on the plasma membrane. I mentioned to you just a bit about the sphingosine 1 phosphate, where it can associate with its receptors for signaling, and these are S1PRs, so this is sphingosine 1 phosphate receptors. There are different kinds of them that act through G-coupled proteins. And for us, they have also, very importantly, they have signaling and bioactive properties. The very well studied one is ceramide. And in general, ceramide is pro-apoptotic, so it induces cell death, growth arrest, and senescence. So this is in general. And most of the time, all these studies are done in cancer cells, and this is why these biologies are the well-studied ones. So I mentioned to you these ceramide synthesis, very important enzymes because they generate different fatty acid chain length containing ceramides that have different properties or signaling properties. I should also say not only signaling properties, but also biophysical properties too, as one can guess. Different carbon numbers on the ceramide structure can affect the membrane structure, specifically packing as well. So here is a slide that I borrow from Tony Futterman. Let's talk about this bottom part. So these ceramide syntheses are highly homologous, and the least conserved one to the others is the SIRS1. That's because SIRS1 actually is lacking this Hox-like domain. All the other ceramide syntheses share this kind of topology. And we are now trying to understand how they are structured. We don't still have the crystal structure of these enzymes, and we are still, we need to learn a lot about these. So throughout the, in the last, I should say, maybe 20 to 25 years, there is a lot of papers published, a lot of research is done. And we know that all these ceramides that are generated by ceramide synthesis have many, many important biological functions. So these can be pathologies, for example, cardiac hypertrophy, in ischemia reperfusion injury, in insulin resistance in response to chemotherapy, and also some other physiological functions, for example, autophagy, mitophagy, ER stress response, and apoptosis. But we still don't know how these enzymes are really well regulated. So when we started this project, we asked this question: are there proteins that can regulate ceramide synthesis via protein-protein interaction? So to answer this, we came up with this kind of a workflow where we could express ceramide synthesis in this HCT116 cells individually with a flag tag, and we do immunoprecipitation of this. We do trypsin digestion and then do a conventional mass spectrometry to identify all the peptides that come down in these immunoprecipitated complexes. And we look at the databases for peptide identification. So here is a heat map representation of the proteins that associate with ceramide synthesis. So on the left, you see ceramide synthesis. This is 1 through 6. We do not have source 3, which is not expressed at high levels in the cell lines that we use anyways. And importantly, certain proteins associated with almost all the ceramide synthesis that we tested. On the other hand, there are certain proteins that associate specifically with certain ceramide synthesis. For example, here, there is a big block of proteins that associate only with SIRS1. And there are some others that you need to pay attention to. And when we look at the distribution of these proteins, we see that these mainly are in the endoplasmic reticulum and also cytoplasm. That's because we think, well, these enzymes are in the endoplasmic reticulum. They span the membrane multiple times. And they also have this exposure to the cytosolic phase of the ER. So therefore, cytosolic proteins can associate with these proteins as well. So these are the biggest components when we look at the cellular distribution. And one protein that we identified that associated with all of them was this enzyme called long-chain fatty acyl-CoA synthase. It's also called ACSL. So we identified ACSL5 to associate with all the ceramide synthesis. So before I show you some data, I want to give a little bit of introduction about these enzymes. So these are very important for de novo lipid synthesis. There are 5 of them in the family, and they are localized to the ER. And their functions, they have functions in fatty acid uptake and, importantly, 3-acylglycerol synthesis. And for us, we are cancer biologists. In addition to being lipid biochemists, these are important for cancer biology, so they are overexpressed in the cancers. And once ACSL5, for example, is downregulated, it induces cancer cell death in many cancer types, including lung, gliomas, and colon. So enzymatically, what they do is they activate a fatty acid. So activation of a fatty acid means that they take the fatty acid and they stick this CoA in a multi-step way in a pyridoxal phosphate-dependent manner. So I mentioned to you that ceramide synthesis utilize fatty acyl-CoA, and then we identified a protein that generates fatty acyl-CoA. We were very excited, and our initial hypothesis was that ACSL5 generated fatty acyl-CoA is utilized by ceramide synthesis for ceramide synthesis. So we wanted to test this hypothesis, but first we wanted to make sure that this interaction that we identified with the mass spectrometry is actually true. So to do this, what we did was we went back to the original samples and looked at the interaction with Western blotting. Here we immunoprecipitate ceramide synthesis and look for ACSL5. We see that it interacts with all the ceramide synthesis that we test. In addition, we also found ACSL1, which is in the family of the long-chain fatty acyl-CoA synthesis. So we could show this interaction with Western blotting in the original samples. Then we repeated these multiple times using a different detergent. So this is not really detergent dependent. We immunoprecipitate ceramide synthesis. ACSL5 is found in the complexes. Then we did the reverse immunoprecipitation where we expressed the ACSL5 with an HA tag. We immunoprecipitate that, and now we can find all the ceramide synthesis come down with that. And then, obviously, if things are interacting, then they would co-localize in the cell. And consistent with their known localization, which is ER, we find that both ACSL5 and SIRS6 here we are looking at do co-localize, and that appears to be on the ER membrane, which is marked by Kalnex. So these binding assays are done in solution, so you need to lyse the cells. So one can criticize saying that there can be interaction happening once you lyse the cells, but this is not specific. To answer this, we have used a technique called proximity ligation assay, where one can detect the interaction of proteins within the cell without lysing them. And if these two proteins associate, which means then they are within 40 nanometers of each other, and we could detect this in a PCR-based manner, and you see all these red dots, they represent interaction. So we could detect this interaction in a proximity ligation assay. And of course, we have also looked, these are in the overexpressed backgrounds. We also looked at the endogenous proteins. We immunoprecipitate endogenous SIRT6 here, and we could find ACSL5. We downregulate SIRT6 using siRNA, and the interaction is much less. So it goes well together, and we did the reciprocal experiments as well. So we were very convinced. We convinced ourselves that indeed ceramide synthesis and ACSL5 do interact using multiple techniques. So then we wanted to identify what portions of these proteins are important for the interaction. So here is a domain structure representation of ACSL5, and here is the localization, which is on the ER. We have generated multiple deletion mutants. I'm not going to go into detail about this. I'll show you this data where we express different mutants, and then we look at the interaction. Very importantly, when we deleted this N-terminus, which is here the first 66 amino acids, and also when we delete the first 88 amino acids, which also includes a transmembrane domain, these two will lose the interaction compared to the wild type with the ceramide synthesis. So this suggested to us that this first N-terminus and the transmembrane domain are important for the interaction. So then what we did was we used, we generated a construct. This is a chimera in which we used this N-terminus, attached it to a GFP, this green fluorescent protein. And then we looked at the interaction of this chimera with the ceramide synthase. And indeed, this chimera can associate with, here we are looking at the SIRS1, they do associate. Then one can ask the question, you are generating a chimera which is targeted to the ER, and it ends up being associated with this ER localized SIRS1. To answer this, we all have also generated this ER targeted GFP specifically, which has a KDEL sequence, so it is retained within the ER. And it is not simply the ER localization that makes the GFP associate, so it is really this N and TM, so this portion of the ACSL5. So here we see that the ER targeted GFP does not associate with SIRS1, but the chimera does. So now we know that this portion of ACSL5 is important for the interaction. So as now we convinced ourselves interaction is real, we were able to identify a specific portion of the ACSL5 that is important for this interaction. Then we wanted to look at the biology. So what happens to the biology? So there are two ways of doing it. One is a biochemical way to look at the ceramides, sphingolipids. The other is the downstream signaling. So first we looked at the ceramides. What happens? So again, I'm going to remind you that we were thinking that ACSL5 would feed into the ceramide generation by producing the substrate for ceramide synthase. But to our surprise, when we downregulated ACSL5, so here you can see with the Western blotting, we were able to see all these increases in almost all of the ceramide species. So this was very, very puzzling, like the Tuticum's riddle, the Sphinx riddle. So here we are looking at the ceramides. Here we are measuring the dihydroceramides. Even the dihydroceramides also are elevated. So then we have repeated this multiple times, obviously. Again, this time we are using ACSL1 as another control. We knock them down using siRNA individually. And in both cases, we see this increase in the ceramides. And importantly, I forgot to mention to you earlier when I was giving you the introduction, but when we use these inhibitors, humanicin B1 or myriosin, which inhibit ceramide synthase and serine palmitoyl transferase respectively. So fumonisin inhibits ceramide synthase, myriosin inhibits the very first enzyme, the SPT. And when we inhibit these enzymes using these inhibitors, and then look at the ceramide accumulation, we could prevent the accumulation of ceramides. So this suggests that indeed the regulation that is done, or the metabolic change that is done with the ACSL siRNAs are through those nodules. So this is within the de novo pathway. So here you can see in the control settings, we see this elevation of ceramides, but when we have the inhibitors, we prevent this. So this is very specific, but it is very puzzling to us. And also, I mentioned to you that ceramide is mostly implicated in the cell death. So these are generally pro-death molecules. Then we looked at in a cancer setting, we have used this 5-FU, 5-fluorouracil, because these are colon cancer cells, and 5-FU is the primary treatment for colon cancer. So here what we did was we downregulated ACSL5 here with the siRNA, you can see, and treated or untreated them. What we see is that ACSL5 downregulation enhances the cell death effect. So here we are looking at the caspase activation, which is a apoptotic marker. This caspase activation is enhanced when ACSL5 is downregulated in the presence of humanis, in the presence of 5-FU. So then on the other hand, now we do the reciprocal experiment, we overexpress ceramide synthesis, because as I said, ceramide synthase generates ceramide which can be pro-death, right? So here we do SIRS1 expression in the presence of ACSL5 siRNA. So there is some cell death with SIRS1 overexpression, but when we combine the two, SIRS1 expression in the presence of SIRS5 siRNA, the caspase activity really shoots through the roof. So it is really enhanced. So the biology really makes sense with the enhanced ceramide accumulation when we inhibit ACSL5, which is indeed, in fact, opposite to what we were thinking, what we were hypothesizing. So this was quite a time. It was very puzzling. So then, this time we did the reciprocal experiments to gain more insight into this. We did the ACSL5 overexpression. So up to now, I showed you inhibition. Now we are overexpressing it. And when we overexpress ACSL5 and measure sphingolipids, now we start seeing these decreases in the sphingolipids. So the reciprocal is also holding true. So biologically, when we overexpress ACSL5, this time now the cells become resistant to the caspase activation, or 5-fluorouracil here again. This again because there is less ceramide, and then there is less apoptosis. So we said, okay, something is happening here. What can be happening? We went to the drawing board. We looked at the structures. This is why it is very important for biologists to know their chemical structures at the same time. So we hypothesized that there can be an O-acylation of ceramide. So here is the ceramide molecule. It has this 1-hydroxy group and the 3-hydroxy group. And we know that ACSL5 generates this fatty acyl-CoA. So we thought when these two come together by an enzyme, we didn't know at the time what the enzyme is, it might generate this compound, this molecule called acylceramide. So here the fatty acyl portion is added to the 1-hydroxy group. So this is 1-O-acylceramide in fact. We said, okay, there could be an acyltransferase that can catalyze this reaction to generate the acylceramide formation. Before investing into it, we measured the abundance of these ceramide species, this 1-O-acylceramides. So this is C16-O-acylceramide in the presence of ACSL5 overexpression. So we overexpress this, and then we look at the generation of this molecule, and indeed when we overexpress, there is increase. And on the other hand, when we do ACSL5 siRNA, now we are blocking this part of the pathway, and then now there is a reduction in the amount of acylceramide. So indeed, ACSL5 does regulate this kind of an activity, which modulates the ceramide levels. So now, I'm cutting it a little bit short here. We suspected that this enzyme called DGAT2, this is diacylglycerol acyltransferase 2, which is involved in the generation of 3-acylglycerols, might be the enzyme. We had many reasons, and there were some hints in the literature coming from the yeast work that was done. So we placed DGAT2. Is this really DGAT2 that is doing this? We have used siRNA to inhibit the DGAT2. siRNA works. And when we inhibit DGAT2, now we are measuring the sphingolipids. We see that the ceramide levels increase. So if we have the pathway like that, so when you inhibit DGAT2, you no longer generate the acylceramide, and then you are backing up the ceramide levels, right? So which is what you see here. And then we also measure the acylceramide levels. When we inhibit DGAT2, indeed we do inhibit the formation of acylceramides. And then we do the reciprocal experiment where we this time overexpress DGAT2. As you can imagine, when you overexpress, you pull the flux towards the acylceramide generation. And indeed, when this enzyme is overexpressed, this acylceramide levels increase. So we were indeed very happy that this seemed to be DGAT2. So what I showed you in this slide is it's happening within the cells. So that doesn't necessarily tell us that this enzyme can have the activity per se to regulate this. Now we are looking at an enzyme activity assay, which is separated on a TLC plate. This is an older technique where you can separate lipids. So here we are overexpressing DGAT2, and we look at the generation of acylceramide from the microsomes after the enzyme activity assay, and indeed we can see the acylceramides. And thankfully there is an inhibitor of this enzyme, DGAT2. When we use the inhibitor in a concentration dependent manner, we can inhibit the acylceramide generation through DGAT2. And then we have also generated a point mutant where it is catalytically inactive, DGAT2. And when we overexpress this and look at in the microsomal activity, the activity is not as high, almost at the background level. So indeed, DGAT2 can catalyze this reaction. So I'm going to go a little bit faster here so we have more time in the end. So we look at the biology, we look at the caspase activation that holds true with the overexpression. I'll go pretty quickly here. So DGAT2 and diacylglycerol generation is shown to be happening in the lipid droplets, and lipid droplets are now gaining a lot of attention as really particular organelles that can support the generation of other lipids for their synthesis and or their storage. So for us, we thought that this acylceramide, which is highly hydrophobic, needed to be stored at a certain place because it's really hydrophobic, and we know in the cells, lipid droplets are the place for the storage of these lipids. So we looked at the localization of these enzymes, where they are happening on the ER when we induce lipid droplet formation with oleate. Now these enzymes end up on the lipid droplet ER interface, which is really the budding of these. We have done this in multiple ways, both using confocal microscopy and also using biochemistry. We were able to isolate the lipid droplets and show the presence of these enzymes, the ACSL5, DGAT2, ceramide synthesis. They are indeed there. And then we will look at the enzymatic activity within the lipid droplets after isolation. When we overexpress DGAT2, there is indeed increased acylceramide generation. So one can say, okay, what is the physiological function of this? Then we looked at the high-fat mice models where we know that there is hepatic steatosis. So these were mice that were fed with a high-fat diet that's enriched in oleate. You can even see here there is these white spots on the liver, and with H&E and oil or red staining, you can really appreciate the accumulation of these lipid droplets in the liver sections. So then we measured the amounts of the acylceramides in the lipid droplets. They are elevated with the high-fat diet. And then in addition to it, there is also increased ceramide accumulation in the lipid droplets too. So one needs to really consider this as a sequestration. I'll explain it to you in a little bit. So in the similar setting, we fed the mice with the inhibitor of this DGAT2, and now we no longer see, or we can inhibit the acylceramide accumulation in the high-fat diet model, which is also very similar in the isolated lipid droplets as well. So here is the model. What we have, what I have shown to you, we have identified this interaction happening with ceramide synthesis, ACSLs, and DGAT2 that I had to go a little bit quickly, where we know that ACSL5 generates the fatty acid CoA, ceramide synthase generates the ceramide, and DGAT2 uses these two to generate the acylceramides. And we know that this interaction is happening on the ER membrane, and this can end up in the lipid droplets, where we see that the acylceramide is color-coded here as this orange, and the complex can also be on the lipid droplet surface. And this is happening in situ on the cells, and in vivo, when the mice are fed a high-fat diet, they have fatty liver. They have increased lipid droplet acylceramide and lipid droplet ceramides as well. And this lipid droplet acylceramide accumulation can be inhibited with the DGAT2 inhibitor that would inhibit this enzyme here. So what are the implications from this? So these results suggested that these very important lipid molecules, ceramide, acylceramide, and the diacylglycerol, triacylglycerol, which is controlled by DGAT, can be balanced together, so there can be crosstalks. And in fact, maybe inefficient acylceramide generation and ceramide packing in the lipid droplets might be a contributing factor for the high-fat diet-induced pathologies, which we now know that indeed it is accumulation of ceramide. And on the other hand, one should really think about the reverse activity, where all acylceramides can be hydrolyzed to generate ceramide once these lipid droplets are mobilized, so that is, all the lipids are digested. So this can be a distinct mechanism of generating ceramide as well. So what would these enzymes be? We do not know. These are a question for the future directions. And also another implication or future study is that can we inhibit DGAT as a combination therapy for anti-cancer treatments? And that is another direction that we are going. And with this work was done when I was at Stony Brook University before my current position, as I mentioned to you. And many people helped with this. At the time, I was working very closely with Dr. Lina Obeid, who passed away, unfortunately. And I'm forever grateful for her mentoring. In addition to Dr. Yusuf Hanun, who is very well known in the sphingolipid field. In fact, one of the first people to show this bioactive functions of the sphingolipids there as well. And I will also tell you a little bit about our laboratory in Richmond. So this is our group in Richmond. We are a very diverse group representing many continents and many different orientations. So we are very happy with this. And again, I'm still collaborating with Yusuf at this point. And our lab has been funded very well, and we are continuing with this project in addition to many other projects. So with this, I'm going to stop the share, and then I'll take your questions if you have. Thank you.
Çok teşekkür ederiz hocam. Dilerseniz ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru cevap bölümüne geçebiliriz.
Tabii geçelim.
Başlıyorum ilk sorumuzda. Is regulation of ceramide sphingomyelin rheostat a targetable approach in chronic diseases?
Evet. Yes. Orada şeyi soruyorlar. Şimdi Türkçe, İngilizce yani fark eder mi nasıl cevap vermem? İngilizce
Yani,
karışık, ben vereyim cevaplarımı. Sen tekrar
hangisinde rahat
sorsana bana
hissediyorsunuz?
şeyi.
Is regulation of ceramide sphingomyelin rheostat a targetable approach in chronic diseases?
Kronik dizilerde belki olmayabilir, bilmiyoruz. Şimdi şöyle bir şey var. Mesela asit sphingomyelinase diye bir enzim var. O şeyde lizozomun içinde oluyor. Eğer bu enzimde bir mutasyon olursa o zaman lysosomal storage disease'ler oluyor. Yani bunlar genelde neuronal disease'ler. Onunla ilgili bazı çalışmalar var. Yani bu bir kronik bir disease olduğu için ondan örnek veriyorum. Onun haricinde yani ben genelde kanserle ilgilendiğim için yani kanser terapiye bakıyoruz. Onunla ilgili de yapılan çalışmalar var. Yani cevabımız aslında evet. What are the biochemical changes in the ceramide-centered metabolism of Sphingol-associated metastatic potential? Şimdi burada da İrem Coşkun Tan'ın sorusu. Herkes görebiliyor mu bu soruyu? Herhalde görüyorlar değil mi? Tekrar
Kusura bakmayın.
Yok sen galiba bir koptun geldin, ben okudum soruyu.
Bilgisayar şey oldu. Tamam, o zaman cevabınızı alalım.
Şimdi cevabımızı verelim. Evet. Yes. Biochemical changes, şöyle bir şey var. Şimdi metastatik potential, genelde metastaz için bu sphingosine 1-fosfate'in metastaz yaptığı biliniyor. Bunu angiogenesis şey yapıyor. Yani kılcal damarların oluşmasında çok etkili. Ve bu metastaz olması için de genelde tümörlerin kılcal damarlar içinden genel sirkülasyonun içine girip öyle metastaz oluyorlar. Onda etkisi var, onu biliyoruz. Onun dışında da yine yapılan çalışmalar var. Bu da hücre zarında, plazma membrane'de ceramide ile ilgili. Yani bu ceramide sphingomyelin'den ceramide'a generate olduğu zaman onun da metastaz ile ilgili şeyleri olduğunu biliyoruz. Kontrol edici etkileri olduğunu biliyoruz.
Cevabınız için çok teşekkür ederiz hocam.
Ben teşekkür ederim.
Bir sonraki sorumuza geçiyorum. Are there specific roles for different ceramides in cell pathobiology? Can they all be associated with diseases?
Evet. There are specific roles for different ceramide species. I can give you an example for that. With respect to, first I'll tell you about the cancer one. When cells upregulate, for example, with chemotherapy treatments, we know that cells upregulate long-chain ceramides. So these can be C16 to C20 ceramides, C16, C18, C20 ceramides, which are known to be pro-apoptotic. So in these cells, if you, let's say, overexpress enzymes that can generate very long-chain sphingolipids, very long-chain ceramides, then you, in a way, overcome the effect. So while long-chain ceramides are pro-apoptotic, very long-chain ones are anti-apoptotic. So this is an example from cancer. And on the other hand, for insulin resistance, for example, we again know that for insulin resistance, we know long-chain ceramides, mainly generated by source 5 and 6, these are C16 ceramides, are the ones that cause the insulin resistance, but not the ceramides from source 2 or source 3, which generate very long-chain ceramides. So there are really differences when it comes to the diseases. In this case, I told you about cancer and diabetes. Thank you. Good question.
Cevabınız için çok teşekkürler hocam. Bir sonraki sorumuza devam ediyorum.
Sure.
Has an anti-metastatic effect of SARS-2 been defined in other cancer types besides ovarian tumors?
Anti-metastatic functions other than I don't think so. I'm not. I do not know. I don't think so. It's most right now. It's what we think is happening.
Cevabınız için teşekkür ederiz hocam.
Sure.
Devam ediyorum sorularımızla. Does systematic de novo ceramide inhibition developed in obesity is the increase in white adipose tissue related to this mechanism?
Ceramide synthesis regular interaction between apoptotic and you didn't read this one, did you? It was a different question.
Yes, it was a different question. I read from the...
Okay, can you tell me one more time? Because I was just looking at this. I'm so sorry about that.
Okay, I'll read it again.
Please. Okay, I see it now. That's perfect. You don't need systemic inhibition developed in obesity. So as far as we know, there is increased ceramide generation from de novo pathway in obesity. It is not the inhibition. As far as the white adipose tissue related mechanisms, I do not know. So this is a very, very big field. So I unfortunately do not know too much about that. I know there are certain studies being done, but I don't think there is any connection at this point with the increase in the white adipose tissue.
Cevabınız için teşekkürler hocam. Bir sonraki sorumuzu okuyorum. Does ceramide synthesis regulate interactions between apoptotic and anti-apoptotic proteins?
Yes, that's a very good question. Ceramide synthesis do regulate, at least from the earlier pioneering experiments from Colombini's group in Maryland. So what they hypothesized that these ceramides can make pores on the mitochondrial membrane, and that can associate with the Bax proteins roles for the cytochrome C release and apoptosis. And I think there are some other examples of this showing that ceramide mediated signaling is important for the BCL2 functions as well. So the BCL2 is anti-apoptotic small protein that is important for apoptosis again. So yes.
Cevabınız için çok teşekkürler hocam. Bir sonraki sorumuza geçiyorum. Is the function of endogenous ceramides produced in mitochondria fully understood, particularly in the context of neuronal dysfunction?
No, we do not know exactly what they do. So this is why it is very exciting to be in this field. We do not know the really what these enzymes, what these ceramides are doing. So it is very critical to identify their interaction partners or their signaling modules, I should put it that way. What are the proteins whose activity is primarily changed by the presence of ceramide within the proximity? These are very important questions. Unfortunately, we do not know. We don't have the full understanding.
Cevabınız için çok teşekkürler hocam. Bir sonraki sorumuzu okuyorum. Bioactive sphingolipids play a role in neurodegenerative diseases. Can profiling of their dysregulation be targeted for disease studies and drug discovery?
Profiling of their dysregulation can be targeted for disease. Role bioactive diseases. So this is a good approach, yes. So one would like to really profile the sphingolipids in the disease setting. Maybe I'll give you an example and then I'll tell you a little bit more about this one. So right now, there is a group in Utah that really proposed the use of serum sphingolipid measurements as a marker for the type 2 diabetic outcomes. So this is very important, and there are correlatory very good studies about that. So for that one, yes, we can do this. It will be great because serum is readily available. Whenever you have the blood test, we have serum, you can test. But on the other hand, when we are talking about the neurodegenerative diseases, it's very hard to get a sample. So one cannot really do a biopsy of a brain or a nerve tissue. So that becomes the limiting factor in this case. So the changes we know are from mainly the mouse or animal studies in the neurodegenerative diseases, or from diseased patients, these changes. So one needs to be careful when we say we are just going to, profile the dysregulation for especially for the neuronal studies. But it is important, don't get me wrong. I hope that we can find certain things that can be checked on the blood that is really regulated by the neuronal functions. So that would be great.
Cevabınız için çok teşekkürler hocam. Sorularımıza devam ediyorum.
Sure.
While silencing ACSL5 increased caspase 3/7 activation, did it cause any change in anti-apoptotic BCL2 expression?
That's a good question. We did not look at it, so that's I don't know if anybody else did. So for us, we were more interested in the sphingolipid metabolism, and the biology is there for us to say that indeed this interaction between ceramide synthase and ACSL5 and the downstream acylceramides are important biologically. This is why we studied the response to 5-FU and apoptosis. But I do not know if there are changes in the BCL2 family. Thanks for explain poor extraction from red blood cells. Poor extraction of sphingolipids. What could explain the poor extraction of sphingolipids from red blood cells? Abdulmajid's question, I do not understand it very well, but at the same time, I know that people do extract lipids from the red blood cells. Maybe their composition is different because these are terminally differentiated cells, right? So they lose their nucleus. Maybe they do not need to have a lot of sphingolipids in them. So this is, structure-function relationship would be important. If there is not much signaling that needs to happen on a red blood cell via sphingolipids, then you wouldn't need the sphingolipids to be there. This is my speculation, obviously, but I do not know the exact answer. So, thank you.
Cevabınız için çok teşekkür ederiz hocam. Sorularımıza devam ediyorum. Can more effective results be obtained in Niemann-Pick's disease with the combined use of SK1A with mTOR inhibitors?
Yes, that's a good question. So in Niemann-Pick disease, this is a lysosomal storage disease for those who do not know, and there are usually mutations in these genes NPC1 and NPC2. These are cholesterol transporters from lysosome to the ER, and others say they can go to plasma membrane too. And when these are mutated, there is cholesterol accumulation within the lysosome, which really messes up a lot of the lipid metabolism and the membranes that one could guess, as cholesterol is one of the main factors giving the membranes their fluidity, right? So I am lucky enough to be involved in a study where we have shown that activation of sphingosine kinases, which generate sphingosine 1-phosphate from sphingosine, can in a way help clear this cholesterol that is accumulated in the lysosomes. This is an approach that we have published, and I think others are also working on. As far as the mTOR pathway involvement, that is also important because we know that the cholesterol accumulation on the lysosome is important for the mTOR activation, right? There are papers from Roberto Zonco's lab at UCSF, I think. But I do not know how one can combine the two at this point. But yes, to answer shortly, yes, both are important. Thank you.
Değerli cevabınız için çok teşekkürler hocam. Bir sonraki sorumuz katılımcılarımızdan geliyor. By analyzing the molecular mechanisms and pathways of sphingolipids in the structure of mitochondria, can the connections on metabolism and cancer cells be resolved?
Yes, that's a good question. We are, when I say we, our group and others that I know in the field try to do these isolations and really look at the sphingolipid metabolism in the mitochondria, not only in the mitochondria, but also in the mitochondria associated membranes. These are really the between the endoplasmic reticulum and the ER. As far as we are trying to understand what is happening and what this metabolism signal to. So yes, we think that we can find the answer, but again, this obviously there are many other things that are dysregulated in cancer. So like many diseases, cancer is a complex disease that one needs to really approach from multiple angles. So we try our best using the sphingolipids as our center of universe, but others obviously do different things, and hopefully we can synergize together. Yes, thank you.
Cevabınız için teşekkür ederiz hocam. Katılımcılarımızdan gelen bir soruyla devam ediyorum. Can lipid metabolism, including sphingolipids, be regulated for the destabilization of brain activation seen in epilepsy and regulation of seizures?
Yes, it can be. In fact, there are cases where, at least in the animal models, we know that this regulation of the sulfatides, these are again part of the myelin membrane, do cause issues with the brain activity and epileptic seizures. So they can be regulated. Yes.
Cevabınız için çok teşekkürler hocam. Sorumuza devam ediyorum. Is it possible to avoid negative consequences and functionally verify metabolite synthesis when sphingolipid metabolism is regulated in psychiatric disorders?
That's a very specific question. I do not know the answer. There might be a connection between the sphingolipids and the psychiatric disorders. But again, it's very hard to sample these before death of the patient. So that's the limiting factor for us. But there might be a connection.
Cevabınız için çok teşekkürler hocam. Bir sonraki sorumuza devam ediyorum. Could the reduction in butyrate production induced by dysbiosis result in downregulation of SARS1 expression in association with upregulation of HDAC expression?
That's a very specific question with the HDACs and SARS1. So I don't know. I didn't read that paper. I need to look at it in detail.
Teşekkür ederiz hocam. Bir sonraki sorumuzla devam ediyorum. Is increased hepatic ceramide levels in alcoholic steatosis associated with hepatocyte damage?
Yes. So there are multiple models for this that I was involved in another study where we looked at with a collaborator, looked at the alcohol-induced steatotic damage. And when one inhibits the ceramide generation through inhibition of ceramide synthesis, one can inhibit the alcohol-induced steatotic damage. So there are studies that are going on related to that. Very good question.
Cevabınız için çok teşekkürler hocam. Bu son sorumuzdu. Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bu bilgi dolu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ederim.
Ben teşekkür ederim. Umarım izleyiciler de beğenmişlerdir. Bana ulaşmak isteyenler e-mailimi bulabilirler. Twitter'da da varım zaten, görüyorsunuz Bioinfocongress'te. Ben teşekkür ederim beni davet ettiğiniz için. Umarım biraz olsun bu sphingolipidlerin önemini anlatabilmişimdir.
Teşekkür ederiz hocam. Sizlere sağlıklı günler diliyorum.
Teşekkür ederim sizlere de. Sağ olun.